Thank you for joining us today. My name is David Stegemeyer. I am the marketing manager for our Gen CRISPR services here at GenScript, and I'm very excited to have Ryan Powell joining us today. Ryan Powell is the CEO at Indie Labs. He is a subject matter expert in biomicrofluidics, and he has been invited to present his work at various events organized by the White House Cancer Moonshot Task Force, the Air Force Office of Scientific Research, Air Force Research Laboratory, National Institutes of Health, among many others. Ryan's been very successful developing Hydropore technology with various forms of support from SOSV, IndieBio, Y Combinator, and among many others. And he was named as a 2022 top 101 biotech CEO in California by Best Startup. Outside of Indie Labs, Ryan mentors, advises, and invests in early stage deep tech startups when he's not out surfing, snowboarding, or traveling. So thank you for joining us today, Ryan.
All right. Yeah, no, thank you very much for the opportunity. I really appreciate it. My name is Ryan Powell. I'm the founder and CEO of Indie Labs. And at Indie Labs, we are developing Hydropore, or instruments for more effective engineered cell therapies. Most people here will already be familiar with this. Engineered cell therapies are important because they can be much more effective than traditional approaches. And then right now, when people make engineered cell therapies, they really have a hard time doing it because the two kind of main means for doing so are viral transduction and electroporation. Viral transduction is kind of slow and expensive, and then electroporation is impractical. It adversely affects the cell state. So despite having all that amazing data already, these engineered cell therapies are held back by their manufacturing methods. That's where Hydropore comes into play. It's a simple shoebox-sized instrument that can simply, effectively, gently, and affordably process cells, allowing them to be engineered. We can do with a single device, you'll see about 55 in this device plate right here. It's about 5 by 10 millimeters. It can process between 1 and 100 million cells. We get a much higher yield of our engineered cells. It's gentle, and eventually we believe healthier cells will lead to better patient outcomes. And then on top of that, we have really low reagent consumption. So, people will use, 10 to 50 times more mRNA than our unit to get similar outcomes. So what we're mainly focused on at Indie Labs is applying Hydropore to autoimmune therapeutics and cancer therapeutics with our collaborators. And we do that primarily with T cells. However, we have done a bunch of different cell types. But T cells are a very democratic cell type, and then regulatory T cells that can help with autoimmune therapeutics are a little bit more specialized. So that allows us to demonstrate our platform demonstrate that our platform is great for rare cell types and your more democratic cell types. You're probably wondering how Hydrocor works. So it's super simple. Inside these tiny little devices are even smaller posts. And then we space them about twice the size of the average cell diameter. And we flow cells past there at about 10 or 20 meters a second, so almost freeway speed. And what that does is it creates these vortices downstream of the posts, and it causes pressure changes and hydrodynamic conditions that permeabilize the cell membrane. So think of yourself at the beach, a wave crashes on you and you feel those fluid forces on your body. We're doing exactly that to the cells in the microscale, and then we're gently pouring their membrane, allowing for delivery. Another important thing is we've already talked to the FDA, and this is not a medical device. So FDA approval is not required for what we're doing. We're tools for biologics manufacturing that goes into your workflows. But because we are conscientious of where we want our instrument to be used, we have developed our instrument in what we call a regulatory ready manner. Everything is ISO 13485 compliant designed, medical grade compressed nitrogen is used, and we use USP class 6 materials across the board. So it's done pretty well. We are in a crowded space, but we do believe that Hydropore is the best product. We have shown that we have improved function of our cells. We have shown that we have reduced reagent consumption relative to our competitors. We have a very small footprint, things the size of a shoebox, and it's super simple to use. I just installed one at a university in Charleston the other day, and it took us less than an hour. We've got an awesome team. We've basically got a small team in our own lab who runs experiments every week, and then we collaborate with universities like UC San Francisco, Stanford, and MUSC, who have like amazing mouse models and constructs that have already developed, so we can prove our platform out for these specific use cases. We've also got some amazing advisors who kind of keep us going down the right path. And then also, if we need some extra help in like biomicrofluidics, we have people like Dino Carlo and Jeff Facer on our advisory board. Indie Labs or Hydropore is protected by a robust patent portfolio, and we have an exclusive supply agreement. Typically, when you manufacture microfluidic devices, it can take up to 12 weeks if you use semiconductor fabrication. And then we have a vendor who will provide them in 3 or 4 days with the same features and quality. So we're pretty excited about that. But now I think it's better if we dive a bit more into the science. So lentiviral and retroviral transduction is cumbersome and expensive, costs a lot of money, takes a lot of time, and then results in therapeutic manufacturing times that are quite lengthy. The only other real commercial alternative right now is electroporation, and it adversely affects cells. Anyone who's electroporated cells knows that it doesn't work very well. It's effective, but not ideal. So we have developed Hydropore. We have had it independently verified for multiple cell types and constructs. We can deliver small molecules, mRNA, and gene editing complexes. And then we are currently working on optimizing our DNA protocols for T cells. But yeah, it's been this platform has been well validated at this point. And it's also a super practical workflow. So if you're used to doing electroporation, there's nothing different in terms of what you're doing. The only difference is you have more time because our buffers we use don't adversely affect the cells, and then you have more flexibility. So if you want to go down to 1 million cells in your processing workflow, you can. If you want to go up to 100 million cells in your processing workflow, you can do that also with the same exact chip. Now, these micro-fabricated devices, they're a bit different than cuvettes. What we have found is that we do have to justify how Hydrocor works when you're using it for research use only. So if you are looking to process a lot of samples with a small number of cells in them, then it's a little bit more expensive. But once you get into the 50 to 100 million cell range, our prices are quite favorable. This is one of my favorite slides, actually. So we took a lot of time to figure this out, but on the left-hand side, you'll notice that the green bar, electroporation or nucleoffection, has really high efficiency. But in the middle on the purple bar, you'll see our second generation platform has about half that. But what you'll realize here in the middle plot is that the purple line is above the green line, which means that our cells within 1 week proliferate much better, and you end up with a higher yield of edited cells despite a lower efficiency. And the reason for that is here on the right. So if you dig in deep and you really want to look at what's going on after electroporation, you'll notice that the cells have very, very low viability within 1 to 3 hours after transfection. And then because we are so much higher than them at that point in time, our cells proliferate much faster. So you get a higher yield. So a lot of people wonder about efficiency and they measure their day 4 viabilities. But if you really dig into it, then you want this really high initial cell viability so you have that higher yield over time. We've also done multi-donor studies. So this is a 5-donor study looking at the differences in TCR knockout efficiency. It was a big experiment, so I do believe the scientists got a little bit tired during donor 1. But for the most part, we seem to be about between 45 and 60% knockout efficiency for TCR, and then we're obviously actively working towards improving that. What we've also done is knock-ins, so using AAV or adeno-associated virus, and we see the similar thing, a higher yield, or about a 1.7x increase in CAR T cells after RNP and AAV delivery for an EGFR-T and CD19 insertion. You can also play around with the numbers a bit. So on the left, we did single and 2-step modifications with the AAV. So basically you mix everything in one Eppendorf tube and you flush it through a device, which is great for more of a manufacturing level thing. But if you're doing R&D and you want to minimize your AAV consumption, then you want to do 2-step where you add in the AAV about half an hour afterwards, and you see the same efficiency. On top of that, we've successfully used Alt-R to modify or to steer things down the HDR pathway, resulting in almost 2x increase in efficiency. So we've shown that this platform can also be improved with things like that. And then nobody wants to use lots of virus, so we've titrated things down, and you don't really see a huge change in a difference in your editing or knock-in efficiencies when you use 4-fold less MOI than standard protocols. So that was a pretty fun experiment. We can reduce the amount of virus, increase your efficiency, and you can use it for research or manufacturing with a simple workflow. Now, the most important thing here is function, right? At the end of the day, if you use a platform for manufacturing therapeutics, like we're hoping to do, then you want your patients to live longer with better outcomes. And so we need to study the function of the CAR T cell killing. And we have a great collaboration with a spin out from MD Anderson that does this in a really cool way. What they do is they put cells, so our CAR T cells and then the target cells, into these micro wells, and they study how they behave together. So here, sorry, wrong button. Here is a picture of our gene-edited CAR T cells in these microwells with our target NOM cells. So the orange ones are the target cells or the model cancer cells, and then the green ones are the CD19-directed CAR T cells. So their job is to go kill those orange cells. And what Cellcore's found was that we got significant synapse formation. So these CAR T cells are functioning. We also got tumor cell killing. So the tumor cells were lysed or effectively killed with our gene-edited CAR T cells and relative to nuclear-affected cells. This is where things get pretty interesting, is what's called serial killing. It's when a CAR T cell goes and kills one target cell and then goes and kills another. So sometimes when a cell will go kill one target cell, it might get, it's called activation-induced death. It'll basically, it takes more of like a martyr or a kamikaze approach, kills a cell, and then it's done. But what you ultimately want is one of these CAR T cells to go in and kill lots of target cells. So this is my favorite part of the study. So we basically showed that serial killing reduces the probability for tumor cells. And around here, around the 3-hour mark, you start to see this red line dip below the blue line. The red line is hydropore, the blue line is electroporated cells. And that's for when there's one effector cell and multiple target cells. But what's even more interesting is that we get what's called reduced activation-induced cell death for Hydrocor CAR T cells. So when there's a 1-to-1, just one target cell and one effector cell in these wells, then we see a bit of a difference between the killing, but within 6 hours. But then on the right-hand side here, you see when there's multiple target cells in each well, we see a significantly improved difference in cell killing. And that's within 6 hours, which is the limit of this assay. You know, there's literature out there showing people are living for 10 years. So a hint of data at this point in time is pretty exciting for us, which means we are working on improving our workflows by reducing the time frame required to manufacture these CAR T cells in the lab. And because we have that really high cell viability and really good proliferation, we can shave that down by about 4 days. And we've compared this to Yescarta, manufacturing time frame based on their peer-reviewed publications, and we believe we could significantly reduce the time frame to manufacture these in these manners. So on top of that, we're also working towards multiplex gene editing in a sequential manner to minimize translocations. So because we have that really high cell viability, we can come back, you know, within 12 to 24 hours and do after our first round of gene editing, and then do a second one. That work is in progress, so you don't see any data there, but we should have it soon. Another one of my favorite data sets is this one. Basically, we're working with the NIH and Stanford on regulatory T cells for graft-versus-host disease, and relative to an electroporation technology, they were able to show almost 100% CAR expression efficiency, but they did work with about, I think it was 95% viability, and they were able to use about 3.8x less mRNA than their standard protocols. So we're in the process of evaluating the function of these regulatory T cells the same way we've had to do with the CAR T cells, and we look forward to showing that data at our next presentation. And then kind of on our technical roadmap is we've got 2 things we're focused on, biology and engineering. Basically, we're teasing out the biology. We're showing these cells have improved function, the CAR T cells and the regulatory CAR T cells. And we're hoping to show we have improved functionality so we can insert even larger genes into the genome. And then we'll look to verify those all the way through to mouse model studies for both autoimmune disorders and cancers. And then on the engineering side, we're super excited. We just got we're working with the NCI on scaling up our device for processing larger volumes of cells. So while the device is great for processing between 1 and 100 million cells, that's not really practical in the clinical and commercial environment. So we are very excited to continue that project, and we'll be making these, pinky nail sized devices a little bit bigger so they can process around, 1 to 3 billion cells. And we also publish all of our data. So if you want to read a bit more about our historical data, you can look into our open source publications on these things, and hopefully we'll have another one out there soon. So thank you very much for your time. I really appreciate it. And if you have any questions, let me know.